All right, so for the last one in our series, we're going to be looking at the blood vessel chapter, our last chapter covered in exam 1. And we're just looking at the exam objectives that are in bold, as they are the potential short answer questions that you could get on the exam. And one thing to note, that doesn't mean that you should ignore all the other exam objectives. Those are certainly important as well. But these specifically are the ones that you should know very well, because not only could you get short answer questions on them, but you could also get your typical multiple choice questions on them as well. So you've got to be able to not only pick out an answer if it's multiple choice related to these, but be able to fill in an answer box, if you will, with the knowledge that you know about how these processes work. Okay? So in terms of our exam resources, don't forget, we have our highlight videos in Panopto. We've got our full-length videos. We've got our PowerPoint handouts. We've got our exam objectives like we're going through, and our vocabulary lists, as well as some of those exercises. All right, so looks like in the blood vessel chapter, again, I have a screen over here. I'm not ignoring you. We've got, I think, 4 different questions in bold that could be short answer questions. The first one is asking you to compare and contrast different blood vessel types. Now this is, again, structure and function. So you need something for each one talking about their anatomy and their physiology, okay? So on this slide, it just gives us a basic layout of the different types of blood vessels that you could choose from. Like we said, oftentimes you only have to name like 5 or something like that if it's a 5-point question, and there are going to be more. So for instance, there are subcategories that you could use as your examples. So the 5 basic types are arteries, arterioles, capillaries, venules, and veins. But in addition, like you can see from this slide, there are 2 different types of arteries, both elastic and muscular. And then with the capillaries, there are 3 different types of capillaries. So really, you have a number of choices about what you discuss if you get this short answer question. So again, you want to be talking about structure and function. One of the basic things that you can talk about in terms of structure is the basic structure of arteries versus capillaries versus veins, right? So arteries are going to have that thicker wall with a thicker tunica media, most notably the muscular layer, that helps to regulate blood pressure and blood flow. All of the larger blood vessels that have that tunica media with the smooth muscle can do that, but because there's so much pressure in the blood in the arteries as it is going away from the heart, it is important that these have a thicker wall to be able to handle that pressure. So veins have the same 3 layers but thinner, and veins have valves which help to get the blood escorted back towards the heart. Because remember, all blood in arteries goes away from the heart, all blood in the veins comes back to the heart. Now capillaries are the simplest of our blood vessels. They're only 1 cell layer thick with that underlying basement membrane. And so their structure and function is definitely there to be able to support exchange with the tissues. They are exchange vessels, and that's why they have the very, very thin walls, okay? So those are our basic ones, and then we have a couple slides that go through and talk about those layers. But after that, we have our elastic arteries, so they have relatively more elastin fibers in them, in all 3 layers of their wall, elastic fibers, and these are pressure reservoirs so that they can stretch out and they have a good elastic recoil to keep that blood going through. Then we have muscular arteries after that. I'm jumping very far, apparently. So there's our elastic, there's our muscular. They have a relatively thicker tunica media where that smooth muscle is. Remember, it's smooth muscle in blood vessels. Cardiac muscle only occurs in the heart. That's going to help to route the blood to the different areas, major areas of the body. Then we get to the arterioles. Again, smaller diameter, but they are helping to dampen that whole pulsing of the blood and reduce the pressure significantly. So by the time we get down to the capillaries, we have a nice, slow, smooth, low pressure blood flow going through. And this table helps to kind of do a structural comparison of those blood vessels. So then we get to the capillaries. Like we said, there are 3 different types of capillaries: continuous and fenestrated and sinusoids. So right there, you can discuss all three. They all have a single layer of endothelial cells, but how leaky those cells are, whether they have holes in them, whether there's holes in the basement membrane as well, has to do with supporting their function. We want them to be leakier, if you will, in certain organs. So, three different types of capillaries. Venules are in essence basically small veins collecting blood, low pressure blood from the capillaries and helping to route it back towards the heart. They do have a porous endothelium so that you can actually get some movement of our immune system white blood cells out of them. And then we get to the veins, and we talked about their structure and function. And they're known as being blood reservoirs because of their kind of increased diameter and the amount of blood that actually stays in them as it's progressing back. Okay, so those are some comparisons of the structural and functional differences of our different blood vessels. From there, the next one is talking about bulk flow. Now, the key thing about bulk flow is that we have to recognize that this is exchange that is occurring in the capillaries. And this is really the exchange of fluids as well as some of the solutes that are in the fluid. Okay? So this is the main slide for a visual that talks about bulk flow. So you have to realize that it's in the capillaries. We said they're exchange vessels. Now ions and water and like respiratory gases, they're going to diffuse on their own through the capillary wall. But again, the vast amount of movement of fluid exchanging with the tissues back and forth into the capillaries is occurring through bulk flow. So you need to understand that as a definition, and then you need to understand what are the forces that cause the water to go out and come back into these systemic capillaries, and really capillaries most anywhere. So there's a variety of forces that are going to be involved, and this slide kind of lays all of those out. In understanding the movement, we have a net movement of fluid out of the capillary at the beginning, going into the tissues. And this is basically called filtration, right? So filtration is where we have fluids dropping through some sort of a porous structure. And then the movement of it is really driven by blood pressure or fluid pressure. So there is a net movement out of the capillary at the beginning that is driven by blood hydrostatic pressure or fluid pressure. And then by the end of the capillary, we're starting to draw these fluids back into the capillary again. And that's mainly due to blood colloid osmotic pressure. Remember, albumin is one of those plasma proteins that helps to create an osmotic gradient to draw water back in. So in essence, that's what's causing the drawing in of water by the end of the capillary. Now, again, there are kind of some lower level opposing forces that are going on as well, but we have a net movement out, a filtration that is occurring at the beginning of the capillary due to blood hydrostatic pressure. And then by the end of the capillary, we have reabsorption of water coming back in, mainly driven by the blood colloid osmotic pressure. Okay? Okay. So the last one talks about blood pressure and how it's regulated by factors that are found out in the body. And this is your main slide that's going to help you with that. So here we see a blood vessel, and you can see both chemicals that may be associated with the blood and with the surrounding area, as well as other types of controls, hormones and things like that, that will affect the diameter of the blood vessel, allowing it to dilate, to allow more blood flow, or constrict. Now, when we're talking about this question, we're talking about blood pressure and blood flow. It's an interesting dynamic, right? Because if we dial open or dilate, if you will, the blood vessel, then that decreases the resistance and there's more blood flow that can go through to that tissue. Meanwhile, if we constrict it, that creates a backup, right? Now there's kind of a backup, like damming up a river or something like that. It creates backup and it increases blood pressure, which is also important. So when you're discussing this one and the various factors that help to regulate blood pressure, you can talk about it as blood pressure or you can talk about it as blood flow. It's fine. Okay. And one of the things that I discuss in lecture is that you should really kind of just think of an example tissue that has these demands based on its activity, like skeletal muscle. And that will help you to get you in mind of thinking of how these different factors could change. So for instance, it's asking about O2 levels and lactic acid and temperature effects on blood pressure and blood flow, etc. So for instance, if you have a very active tissue and you see that there's a drop in oxygen levels because it's being used in aerobic respiration, then the response by the blood vessels is to dilate so we can get more blood there, deliver more oxygen to that active tissue, right? So it's going to increase blood flow. And in theory, it could decrease blood pressure, but you're going to get more blood to that particular tissue that needs it. So a drop in oxygen is going to increase blood flow. An increase in carbon dioxide, because that tissue is very active, that's going to increase blood flow. A drop in pH, whether it's due to lactic acid or not, shows that there is an increase in activity of the tissue, so we need to increase blood flow to it. Remember, we'll talk about carbon dioxide production and how, in essence, it affects pH because it gets converted into bicarbonate ion. So the ion that we have there, as we have that conversion of it, is going to affect pH. It's going to make it more acidic. So the more carbon dioxide that is produced, the more acidic that fluid becomes. So you can see how those are linked. An increase in our potassium in that surrounding area, as we saw, depolarization, repolarization, that type of thing, also indicates that there is an active tissue there. So what we're seeing with these different factors is really all you need to do is say, if this factor goes up or down, what does that do to blood flow to the tissue, right? So you can just say, as this goes up, this goes down. As this goes up, etc., right? Okay. All right. So other things that we were seeing, what other things were we seeing? Temperature, right? So as temperature goes up, that's an indication of an active tissue. So the blood vessel is going to dilate as well. What else do we have? We had some hormones there. Antidiuretic hormone is working to increase blood pressure. So that's going to cause constriction. Atrial natriuretic peptide, AMP, is looking to decrease blood pressure, so it's going to dilate these blood vessels instead. We're trying to reduce blood pressure and even maybe get rid of some of the fluid out of the system. Epinephrine, we know, is going to increase not only the constriction of the heart, but it's going to constrict these blood vessels as well. And we see that over on the left-hand side here, at least for the alpha receptors. What else? The RAS mechanism, the renin-angiotensin-aldosterone system, that one is looking to increase blood pressure as well. And so you see angiotensin over there on the right as part of the RAS mechanism. That works to constrict our blood vessels because we're looking to increase blood pressure for whatever reason. Nitric oxide tends to dilate. You should have it. You see it up here. I'm just looking over the list. And it looks like basically that's it. Now, again, you won't be asked all the different factors, but you'll be asked a list of them, and you'll have to choose some and describe what the effect is on blood vessel diameter and blood flow, etc. Okay, the last one has you compare the structural and functional differences between the adult and fetal circulation. And so that one comes into play in our last few slides. This one is the main one that you can use. It shows 1, 2, 3, 4, 5 different comparisons. But of course, down here at the bottom, we can see in the fetal circulation shown on the left that, to be sure, having a placenta is going to be a difference in the circulation between the fetal circulation and the adult circulation. So for these structures, you would need to describe why it's there. And then in the adult, what it becomes. Is it totally gone, right? So the afterbirth is the placenta. It's removed after the birth of the baby. And so there's no placenta and no exchange with the placenta in the adult. That's a good way to describe a comparison there. Let's look at these other ones. So ductus arteriosus in the fetal circulation is going to be a shortcut between the arch of the aorta and the pulmonary circulation. So in this, it allows blood flow to go directly from the arch of the aorta and the pulmonary circulation to be connected so it can go out to the body faster. Whereas in the adult, that becomes the ligamentum arteriosum, just a remnant of connective tissue. So there's no blood flow there anymore. Foramen ovale as a whole, connecting the two atria, again, getting blood flow to the systemic circuit that much faster because we've already exchanged blood here at the placenta, exchanged oxygen in the blood down here at the placenta. So let's get it out to the systemic part of our circulatory system faster. So it's a hole there. And then in the adult, it becomes the fossa ovalis, right? Like we saw on the heart models. Ductus venosus is a shortcut that links our umbilical vein to the inferior vena cava so that we can get this highly oxygenated and nutrient-laden blood from the placenta back into the fetal circulation. And that becomes the ligamentum arteriosum in the adult, which is just connective tissue, so no function, but connected to the navel that we know, the belly button. And then finally, we have the umbilical arteries, which would be taking fetal blood out to the placenta to do oxygen and nutrient and waste product exchange. And ultimately, that becomes the medial umbilical ligaments, again, with no circulatory function. Okay? So that's how we would compare our circulation of the fetus and the adult. All right. So that should do it in terms of your exam objectives. Again, I just wanted to get these out there to you just because this is our first lecture exam and you're trying to kind of get your footing and figure out how it's going to be. And I hope that helps. So come see me or email me if you have any questions.